Hello everyone. I made this video so that I can show you how to use a program called H++ from Virginia Tech, and the purpose of this program is so that we can explore titratable groups in proteins. So the first thing that you're going to want to do is to log on to the H++ site. So if you just search H++ and Virginia Tech, that should come right up. And what you would like to do is you need to create an account, and it's free. And the reason that you want to do this is because if you create an account, I can go back here and I can hit where it says view submissions. So these are the jobs that I've done in H++ since I started using this program. So you can see that some of them are recent, but I did attend a workshop given by Dr. Ellis Bell at the University of San Diego in the summer of 2020 when we were, as you might remember, doing a lot of things that were remote. So the structure that I processed as part of that workshop is still here. So if you create an account, then you'll be able to access any of your structures. So if you want to process a structure, what you should do is click on Process a Structure, and then we can choose a file. And the file that I want to choose is, I'm going to go to my Dropbox and go to H++, and I'm going to use the file that I alpha folded. So that's the P40925 variant 3, so that's the human MDH, cytoplasmic MDH1, and I'm going to use the alpha folded. So what you're going to do is you're going to use your mutant alpha folded protein. So when you hit process file, then it will say that this protein has a lot of titratable groups, which it does, and it estimates that the runtime will be about 5 minutes. So this is normally a fast protein. So for us, this is actually a little bit on the slow side. So if I scroll down here, I'm going to leave the salinity and the internal dielectric on the defaults. The external dielectric, that's the value for water, so I'm also going to leave that as the default. And I'm going to also leave the pH as a default. I'm going to leave all of these as defaults too. I'm going to change the solvent box to cubic and leave everything else as a default, and then I'm going to hit process. So since this is likely to take a few minutes, what I'm going to do is I'm going to stop the recording, and then I will start it back up again once we have our results. Okay, so we're back. That actually didn't take the 5 minutes that it said that it was going to. We can view our results. So it's actually fairly quick. So if I hit view results, then what I'm given is the amino terminal and all of the titratable amino acid residues in my protein and my carboxyl terminal. So one of the things that you can do is you can view the titration curve, because we're essentially doing virtual titrations of all of these amino acids to obtain these values. So if I click on, for example, the histidine 205, and I chose that because the histidine 205 for the cytoplasmic malate dehydrogenase is one of the active site residues, and if you might remember, it is protonated, and we can see that the value here is 7. So again, if you look at the midpoint of the titration curve, you can see that 0.5 and drop a line down, and there it is. So we do virtual titrations of all of these amino acid residues, and the pKa values of the side chains are determined from these titration curves. So we have some other information here too. So we can see that the isoelectric point of this protein is 8.4. The total charge at pH 6.5, of course, is going to be 2. And we have these files here that we can download that will give us all of the pKa's of these values. So if I click on this one, what's nice about the way they name this is that it has the salinity and the dielectric constant and the pH and the name of your file, so you know exactly what it is. So if I click on it, I'm given a table of values, and I've already done this, so I'll just show you how to do this. So what you should do is you should go to your browser, and then you should print this to a PDF. I have already done that, so I'm going to go back So that's one of the files, and then this is another file right here. So this file will give you all of the residues plus the amino acid side chains that influence the pKa value. So I'll go over that in a minute. And then the third file that I like to download is this PDB file. So this is a PDB structure that has all of the amino acid side chains in their predicted proteination state under the conditions under which I ran this. So that means that you can go into Pymol, and then you can look at those proteination states. Okay, so I've already done this, so I'm going to go back here. First, I'm going to show you this slide here because I just sort of marvel at its completion. So the credit here goes to Dr. Ellis Bell at the University of San Diego, who put this marvelous slide together in terms of why are the pKa's of these titratable groups so important. So I'm going to focus right here. And tell you that the pKa of a titratable group is governed by its local environment. So the nearby side chains contribute to local polarity and charge environment. So are these, if you look at a specific amino acid side chain, is it near a positively charged amino acid or a negatively charged amino acid? So depending on who its nearest neighbors are, there will be an impact of the pKa of the specific group that we're looking at. But it's not just charge residues, because we also have polarity and other types of interactions that impact the local environment. So the data we get from H++ can tell us all about this. So here is the first PDF that I downloaded. You can, if you want to, export this to Microsoft Excel. I'm going to leave this in the PDF file for now. I think it's maybe a little bit easier to navigate, but that is something that you can do. So the way that this works is this is the first file, and it lists all of my titratable amino acid residues and their positions on the protein. And the first pK value is the pK value that the program calculates as if it is not influenced at all by its nearest neighbors. But so the second one shows again, we're doing this virtual titration, and these are values that take into account the nearest neighbors. So we're going to want to look at this second column here. The other thing to notice about H++ is that it calculates values between 0 and 12. So if a pK value is less than 0 or greater than 12, it will just indicate that. So I'm just going to go to just a couple amino acid residues just to for illustration. So here, this is the histidine 205, and you notice that when you take into account the nearest neighbors, that the pKa goes up by 2 units. The pKa of this histidine is 7.02. So I'm interested in that because it's a active site amino acid residue, and I'm also interested in this aspartate, which I notice goes down quite a bit. So we can, if we look at these pK values, we can see that at the pH that we chose, that the histidine is protonated and the aspartate is definitely not protonated. And if you go back and look at the mechanism of the dehydrogenase enzyme, you can see that the asp not being protonated and the hist being protonated are essential to its mechanism. One of the things I did want to point out is this shows HID instead of HIS. So I went back to the protein data bank, and where am I? Okay, so this is from the protein data bank. So histidines can be represented by different ways. So our friends of the protein data bank say that histidines are tricky. They have 3 possible proteination states. So HID so this is the delta tautomer. So histidine exists in 2 different tautomers, delta and epsilon. And so in the delta configuration, the proton is found on the delta nitrogen, and in the epsilon tautomer, the hydrogen is found on the epsilon nitrogen. HIP means protonated, so so the HIP is the protonated residue. So here it says HID, so that's the delta tautomer. All right, so if I go back here, what I want to do now is I want to go back here to this document. You can see it's quite long, but what it does is it lists every single one of the titratable residues. So up on top here is the site. So the site of interest is listed first, and followed by a list of neighbors with strong contributions. So there might be neighbors that make strong contributions or not. The first number is the background contributions from non-titratable groups, and the last one is the interaction with titratable groups. So if I go to the HIS 205 that I was looking at before, remember we said that it's PK, the pKa value of the histidine was 7.02. Oh, I think I overshot this. Where am I? Okay, so okay, so there it is. So there's our histidine 205, and what I see here is that there are so many contributors to this pKa that I can't even list them on this one page. And that makes sense because the histidine, this is an active site amino acid residue. Its ability to be protonated is very much contingent on its enzyme activity. So if you look at this, so the first is the contributions to the, how did I get to the 7.02 pKa? So these are all the contributions right here. Remember, these are the non-titratable groups. So if you look over here, you can see that there are a lot of zeros. So if you look at valine and glycine and asparagine and proline, those aren't titratable groups. So they might have electronic or other kinds of effects, but they're not going to have any kinds of charge effects because they're not titratable. So if I look at the histidine, we can see that if we have negatively charged groups like an aspartate, cysteine. Now here, that's our aspartate 177, which is part of the active site. So look how huge that is. So these values are all positive, which means these amino acids contribute to the histidine having a high pKa. So the histidine has a high pKa. So a high pKa means that the histidine is going to be protonated. So the higher the pKa, the greater the percentage of the histidines that are going to be protonated at any given pH. So it's interesting that our 177 contributes greatly to ensuring that our histidine is positively charged. But if you look at other positively charged amino acids, like if you look at arginine, other histidines, you see that those are negatively charged. And again, for those positively charged amino acids, they would prefer that our histidine not be protonated because that would lead to repulsive forces. But we see that the negative contributors win out, and we have a histidine with a high positive charge. If we go back up and look at our aspartate 177, so that is up here. Okay, let's see, where is it? So again, more contributors than I can have right here. So remember, the aspartate was listed by H++ as less than less than 0. So I'm not sure about that for a calculation, but anyway, a very low pKa value means that the aspartate is definitely deprotonated and negatively charged, and for the mechanism it needs to be so. And again, if you look at the kind of the contributors, so you can see that other positively charged amino acids like arginine and histidine will have a large contribution to ensuring that that pKr value of the side chain is very low. And we can even look at our histidine right here. So again, we see a very large negative contribution, which means that the histidine has a pKa lowering effect of the aspartate. And of course, other negatively charged amino acids are going to have the opposite effect. So I've lost my place already. That's 175. Okay, so if we look at other negatively charged amino acids like where am I? Okay, let me scroll down a little bit so I can see if I can find some. Okay, so here's a glutamate, for example. So again, that's going to want to inch the pKr value up, just because if you had a negatively charged glutamate and a negatively charged aspartate, that would be a repulsive charge. But again, all of these contributors, the contributors that decrease that have the pKa lowering effect for the aspartate win out, and the aspartate has a low pKa. So in our wild type enzyme, we have histidine, which is positively charged, protonated, and aspartate, which is negatively charged, deprotonated, and that is essential to the enzyme mechanism. So what you're going to want to do for your mutation is, I remember that one of you, I don't have all of my mutations in front of me, but I remember that one of you was looking at the 110 position. So let me go up here and just use that as a quick example. So if you modified, a lot of you carried out mutations where you took a titratable residue and mutated it to a non-titratable. Okay, so here's our arginine 110. So if you mutated the arginine 110 to something that was non-titratable, it would be interesting for you to look at these data. So again, here, this is as we expect. So ARG110, if I go back to this sheet right here, so that was my first file. So ARG110, its pK, where am I? Yeah, so its pKa value is greater than 12. Again, not unexpected for arginine. So again, we would expect that negatively charged amino acids like glutamate and aspartate would contribute to that value being high, because that means that the aspartate and glutamate are negatively charged and prefer to act with a positively charged arginine, whereas other positively charged amino acids like the histidine and arginine would have the opposite effect. So if you mutated this residue, it would be interesting for you to look and see, to compare from the wild type to your mutant to see how these pKa values are different. And the other thing that would be interesting for you to do is to look for your mutated amino acid and see how it contributes to the pKa values of other select amino acid residues. So this is 33 pages long, so you don't have to look through all of it. But see if you can find some significant contributors, say differences of maybe 0.5 pKa units or greater. So I wanted to show you some of the projects that were shared with us. So these are projects that students at the University of San Diego did during the pandemic when they were unable to work in the lab. And they did quite a lot of work with H++. What we're going to do, and sort of following in their footsteps, is we're going to compare our mutant and wild type protein with respect to the local environment. So one of the things that the students did is that there is really no object to how many mutants you can make if you're doing virtually. So these particular students, what they did was that they made 6 different mutations. So this student chose arginine 130, and I believe they might have been working with the watermelon enzyme. So their positions are different from ours because we're using the human cytosolic MDH. So they looked at arginine 130 and they mutated the arginine to allyl, phe, lys, glin, or glue. And then what they did is they looked at the differences in the pKa's between the wild type and the I'm getting a lot of pop-ups here, so sorry about that. I'll try and move my cursor so I don't do that. That they looked at selected residues, which you see over here on the x-axis, and looked at the differences in the pKa. So what they did is they took wild type minus mutant. So the pKa value of that residue in the wild type subtract from that the pKa and the mutant, and you can see do the pKa values go up or down depending on the mutation. So you won't have as much data because you only have one mutant to work with, but it would be interesting in your paper if you could have a graph graph like this and show the differences in selected amino acid residues and look at the differences in the pKa values that you get when you carry out your mutation. The other projects that the University of San Diego students did is that you can choose various residues. Now here, histidine is a good choice. And they looked at the impact of the local dielectric constant. So we set ours at the defaults, but if you could run a number of H++ virtual experiments from 0 to 100 and keep the salinity constant, and you can see that for some of the histidines there were great changes in pKa values depending on the local dielectric constant, and some of them that there weren't. Same thing over here, that the salinity, again, if you go from 0 to 1.2, you could look at the influences of the salinity on the pKa value. So lastly, what I want to show you is that the other file that you could download is the PDB file. And so this is the PDB file that I downloaded. So I'm just going to hit S for sequence, and I'm going to look at the active site residue since I was looking at that before. And if I look at the ASP 177 and the HIS 205, so I'm just going to select those. And then I'm going to show these as side chain sticks, and then I'm going to color these by element in a different color so I can see them. So what you can see with it, if I zoom in on here, that the pi mole, which normally doesn't show hydrogens if you don't tell it to, you can see that it shows the histidine completely protonated. So these little white sticks right here are protons using the CPK coloring. So we can see that our histidine is protonated. And if we look at our aspartate, let's see if I can zoom in on that. So if I look at the aspartate, then there's the CH2, and then there's the C double bond O, and there's the oxygen right here. And you can see that the aspartate is not protonated. And that was what the data had showed us. So if you're interested in various amino acid residues, you can open your PDB file generated by H++ and see what the state of the protonation is. So I'm going to go back to our H++, and again, if you will go under process the structure right here, there are lots of different things that you can do, lots of different virtual experiments that you can do with the wild type and with the mutant, and it would be very interesting to see what the differences are. So that's what I have for you today. Thank you for your attention, and I hope you enjoy working with this program.